It was... That does seem to be a theme through all these podcasts.
I don't know what you mean, Don.
Yeah.
Welcome again to Free Associations from the Boston University School of Public Health, the Public Health and Medical Journal Club podcast for anyone who is as confused by the latest health study as I am by self-checkout machines at the grocery store. Can you guys deal with those?
Or the parking meters in Medford, Massachusetts.
I can't. Any of those
can't figure
things.
them out.
What is going on with Demi Moore these days?
The whole what?
Never, sorry.
Lost me there. I'm Matt Fox. I am professor of epidemiology and global health here at the Boston University School of Public Health, and I am in the studio with Dawn Thea and Chris Gill from the Department of Global Health.
Hey, Matt.
Howdy.
And we are here in the Boston University Godly studio. So, guys, how do you feel about exchanging things? Do you guys return things when you buy them online? And
No, I like to simply de-access.
Oh.
I don't like getting anything in return. I just want to get rid of stuff.
How do you feel about populations? You like them?
Oh, yeah.
And
I see where this
how
is
do you feel
going.
about health? You into that?
I'm into that.
Because I have a place where you can get all three, which is the Population Health Exchange. That is Boston University School of Public Health's resource hub for lifelong learning. Find out more at www.pophealthex.org, where you will find this podcast as well as many other population health learning programs and tools.
It's kind of like a giant consignment store.
It's kind of like a giant consignment store, but much, much better. So, and as a reminder to all of our listeners, if you could go onto iTunes or any other major podcast site and give us a rating, give us a review, we would really appreciate that because it would help people to find us. So now on to the show. So today in our first segment, which is our Journal Club segment, we are going to look at a study which looked at the effects of a vaccine that was, I'm going to say designed to prevent tuberculosis, but I don't know if it was specifically designed for that, but a vaccine that is used for tuberculosis, whether that can help with type 1 diabetes. Then in the second part of the podcast, which is our deep dive, we're going to talk about something called the Loss of Confidence Project. And then in our third segment, which is our amazing and amusing, we will get into some things that have us chuckling to ourselves, or Don will say something so ridiculous that we have to spend hours and hours editing it out because I simply cannot continue. Before we do, I want to say one thank you to all the people. I was at the Society for Epidemiologic Research meeting a couple weeks back, and I got a lot of people who came up to me and said that they really enjoy the show. So to those of you who are out there listening, we want to say we really appreciate it. Please continue to give us your feedback. So
You got no bad feedback?
I'm not going to say I got none.
Do tell.
But that's more of an off-the-air kind of thing. So let's get into segment one. So in segment one, we are going to talk about an article that looks at whether or not a vaccine that is used in much of the world, though we don't use it here in the United States, to prevent tuberculosis can be used to treat type 1 diabetes. This was a study which is published in a journal that I didn't really know, which is NPJ, Nature Partner Journals Vaccines, NPJ Vaccines. And the first author, Don, you're going to have to help me out with this one because I can't pronounce it.
You're on your own.
W.M. Kutriber,
Yeah,
let's say, of the immunology laboratories at Mass General Hospital and Harvard Medical School. The study was titled Long-Term Reduction in Hyperglycemia in Advanced Type 1 Diabetes, the Value of Induced Aerobic Glycosis with BCG. Glycolysis with BCG vaccination. So we need a big board of all the different words that I have butchered over the course of this podcast, and then I could just keep recycling. So let me give you some of the headlines on this one. So Breitbart News, our favorite.
Oh, please. Come on.
I did that for you.
Oh.
Breitbart says, genetic vaccine reverses effect of type 1 diabetes. Time magazine says
It's not a genetic vaccine.
It's not a genetic vaccine, so they got that pretty wrong.
Sorry, did I say genetic?
You did.
Sorry, generic.
Oh.
Generic.
I wouldn't say
Sorry.
it's a generic vaccine either, but okay, whatever.
Time magazine says there's hope for a vaccine to prevent type 1 diabetes. Fortune says tuberculosis vaccine could reverse type 1 diabetes, studies show. NBC News says, can a TB vaccine help treat diabetes? And Newsweek says type 1 diabetes, tuberculosis vaccine could offer, quote, major advancement, end quote, in treatment. So there you go. So Don, let me start with you as always. Can you explain this in language that I can understand,
Oy.
as my mother would always say in relation to me talking to her about computers? Can you explain this in language that I can understand? Because this was of all the studies we have ever done, the most complicated one I have ever encountered.
Yeah, this was a complicated one. It is different than a lot of the studies that we've covered before because it doesn't have a very heavy epidemiologic component. It's really a combination of two studies. One, it's got a clinical part, and it's got a laboratory part. But let me step back from that and set the stage a little bit. We were really excited about this study because it really is a study that proposed a complete paradigm shift in two respects. One was that there was a vaccine that could potentially have an effect on type 1 diabetes, and that the effect that they thought they were looking the reason for the effect that they observed that they were looking for ended up not being the effect that they had anticipated, and it took them in a different direction, and it brought them to a conclusion that was completely unexpected. So it's sort of a mystery story. But before I start, let me distinguish for the listener. There's type 1 diabetes, which is the kind of diabetes that is insulin dependent, and type 2 diabetes is a kind of diabetes that is not necessarily insulin dependent. The second is more associated with being overweight. The former is more associated with people that get diabetes when they're young. So it's a lack of insulin.
And we're talking about type 1 diabetes here?
In this study, we're talking about type 1 diabetes. Let me also talk about BCG. BCG is a vaccine, Matt, as you said, used to be used in the United States, but hasn't been since about the 1980s. Likewise, in Europe, it was sort of phased out in the mid-80s. It was originally developed for tuberculosis, but has been found to be not as efficacious against tuberculosis as previously thought. And because tuberculosis became less of a problem in the developed world, it was sort of phased out. That said, there has been a tremendous amount of work in the last 10 years, and this is literature that I wasn't really familiar with, in the last 10 years about the effect of BCG in other settings. And to not get into too much detail, the findings are really quite intriguing. There's a study that was done a couple of years ago in Denmark, which seemed to indicate that if children got this vaccine when they were young, either in first grade or before, that when they followed them into middle age and later middle age, they determined that there was a lower all-cause mortality for reasons that are not completely clear. They also identified that people who get this vaccine seem to have improvements in multiple sclerosis, and there's some evidence to suggest that the same thing is true with rheumatoid arthritis. And the idea was that it was in part due to the vaccine's effect on decreasing a state of autoimmunity, where the immune system's kind of revved up. They've also noticed that animals injected with BCG have been shown to have resistance to certain infections such as Staph aureus, herpes, salmonella, listeria, and that there is some suggestion that there is also this mortality effect in humans. Now, how did they get to diabetes? There's been laboratory work that on mice that have in which they induce diabetes. That BCG seems to, in fact, decrease the mice's need for insulin, and their theory was that the BCG vaccine altered the immune system such that the mice who were in a state of autoimmunity, where their immune system was sort of eating up the cells that produce insulin, the islet cells, were no longer under attack after the vaccine, and the mice could regenerate those cells, so they could produce insulin again. So they asked the question, can we demonstrate that in humans? And they happened to also be involved in a study from 2000, a number of years ago, I can't remember exactly when the first study was, where they gave 2 doses of BCG to type 1 diabetics, and they looked at the effect over about, I think it was 20 weeks, and they didn't see any effect, and they just kind of forgot about that, but kept
effect in terms of blood sugar.
Blood sugar, yeah. And the way they're measuring the blood sugars, they're looking at something called hemoglobin A1C, which is really kind of caramelized hemoglobin in your body. If your blood sugar is up for a long period of time, your hemoglobin becomes caramelized, and that's reflected in this measure. The higher it is, the worse your hyperglycemic control is. And so they didn't see anything early on, but when they came back and looked at these same people 3 and 5 years later, they noticed that their blood sugar was in better control. Now, these are type 1 diabetics, so they're getting insulin, and their insulin is being controlled by themselves. But when they looked at this group who had gotten BCG previously and compared it to a group that had gotten a placebo, albeit a small group, and a group that were just type 1 diabetics who were normally treating their own disease, they found that there was a substantial difference, between 10 and 20% improvement in that hemoglobin A1C measure. And so they thought, all right, well, this is interesting. Let's figure out whether it's the same mechanism in humans as it is in mice. And so they did a whole bunch of bench experiments. And what the first thing that they did is they tried to find out whether the humans who had gotten BCG were now making insulin that they previously had not been making. And the one laboratory measure to look at that, which is a little piece of a protein that's cleaved off the insulin molecule as it's produced by the pancreas in humans, should go up if their islets are being reconstituted. They looked for that, they didn't find it. So that kind of ruled out the idea that the BCG had induced their pancreas to now produce insulin. Mm-hmm. Scratched their heads a little bit. They couldn't really quite figure it out. And then this is where the paper gets really complicated, and they looked at a whole bunch of different things. But to kind of make a long story short, what they did is that they looked at the white blood cells that they took from these different groups, the type 1 diabetics who had gotten the vaccine, the ones who had gotten a placebo, and the ones who got neither. And they started to ask questions about how do these white blood cells handle glucose. And what they did was that they established the fact that these white blood cells handle glucose differently. And this is 3 and 5 years after they got these 2 doses of BCG. They handle glucose differently. The way normal people handle glucose is through a process that's highly efficient, takes the glucose molecule and makes it into energy packets, which is absolutely fundamental and critical for all mammalian life, really, almost all life. And the way we do it is a highly efficient process called oxidative phosphorylation. And when they asked at the bench whether, in fact, these cells are still generating energy, using glucose to generate energy in the same way, they found that, in fact, they weren't. What they were doing was that they were using an alternative pathway of generating energy, which is slightly less efficient than the normal way. And because it's less efficient, it requires more glucose. So this new means of producing energy that was seemingly induced by this BCG is called aerobic glycolysis. Hence,
Anaerobic.
Aerobic glycolysis. It's aerobic glycolysis because it's using oxygen. So it's the kind of, it would be anaerobic glycolysis in the absence of a sufficient amount of oxygen. But because there is a sufficient amount of oxygen, it's aerobic glycolysis, which is more efficient than anaerobic. Anaerobic is really inefficient, which is why you get the buildup of lactic acid and why your muscles burn when you don't, when you exercise too much. So they seem to show that there is this permanent transfer of energy utilization, glucose utilization to produce energy. It sort of takes it out of the mitochondria, which is the part of the cell. It's an organelle of the cell that's the efficient place where oxidative phosphorylation happens, and transported it into sort of the cytosol, sort of the regular fluid in the cell outside of the mitochondria. And the end result is that, in essence, the entire system is using glucose less efficiently, therefore consuming more glucose, pulling more glucose out of the blood, using it in the cell in a less efficient way to generate the same amount of energy. So it's sort of like almost down-regulating the body's efficiency in using energy. And what it does is it takes more glucose out of the blood, sticks it in the cells to generate energy.
It sounds completely fascinating.
It's a complete paradigm shift in our understanding of basic biochemistry. Chris and I had to go back to the biochemistry tables and manuals and say, is this really happening? This is against the central dogma of what we learned in school. It's really very, very remarkable.
There are a lot of words in here I don't understand, a lot of things that you said that were outside of my understanding. Whenever I see the word Krebs cycle, I assume this is a paper that I cannot read. But I can say that I got through it enough to get the sort of the general premise that essentially we've got the vaccine is inducing something to happen in the body that is changing the way that the body uses glucose, I guess you're saying.
More efficiently, so therefore consuming more, therefore emptying the blood.
Which essentially leads to disease control.
Right.
Okay. Chris, what's your...
I loved having to go back and study the Krebs cycle again.
I'm sure you did.
Which, I guess, how many times in our lives have we done that? I'm thinking back to high school biology, college biology, medical school.
I only studied it just
before high school biology.
Just before the exam when I had to cram for it.
I don't ever remember this.
I was just thrilled that I remembered that there was such a thing as the pentose phosphate shunt.
Okay.
And I did not realize
you're
that this
showing
is...
off now, Chris.
Yeah, you really are.
I'm looking at the creation of phosphoryose.
Okay.
Stop.
You're beyond me. Go give us your critique here.
I thought this was a fascinating study. I mean, we'll say right up front that this is definitely one of the ones where I think I'm going to come up and say the more research is needed and they need a bigger follow-on study.
Oh, absolutely.
I mean, because the numbers that they're talking about who they followed for these 5 or 8 years was really a very small number.
How small are we talking? Let's get into it.
Well, they started with 52, and then they were somewhat vague about how they did the randomization. But then when they finally got down to the long term,
the
we're down to 6 and 6,
follow-up, which is what we need.
6 people and 6 people.
I thought it was 8 and 8, but fair enough. It's a very small number.
We'll take the average and say it was 7, but it was a real small number.
It's a real small number.
If we were approaching this as a traditional epidemiologic study, we would be hammering in the nails right and left here.
Yeah.
But this is not really an epidemiologic study. This is really a biochemistry study, in which there was a human intervention. And so I think the way we think about this changes a little bit because so much of the evidence rests on the biochemistry and the immunology that they're trying to present. And Don, I thought you did a fantastic job explaining that.
Thanks, Chris.
I guess I would just like to recapitulate that the authors went into this with 3 theories about how the BCG sugar reduction might work. One is that the BCG in some way renders the pancreas able to regenerate its islet cells and create more insulin. And as Don said, they looked at C-reactive peptides, and they did not find that after stimulating the insulin that there was insulin being secreted by the pancreas.
That's the little piece that breaks off of the insulin as it's being produced in the pancreas, and that you can measure. So you couldn't measure the regular insulin because the stuff that they take in the needle is identical to the stuff that their pancreas makes. So you have to look at the byproduct.
Exactly. So they have to look at the C-peptide instead. So that's a marker that this is human-derived insulin. And it wasn't coming out, or at least it was not coming out in very appreciable numbers. So the hypothesis number 1, wrong. It's not that the pancreas has regrown in the way that the mouse pancreas did. So the second hypothesis is that perhaps the insulin that's there is just being more efficiently used. And so then they went through a series of experiments to look for markers of increased efficiency of insulin effect. And they were able to reject that as well. And so then the third hypothesis really by default is other. And that's where really got interested in, and they looked into, well, what could be other ways in which the metabolism of glucose is altered. And that's where they got into this aerobic, glycolysis and the use of these accessory pathways. And they were able to show that, there was a significant downregulation of the downstream utilization of the Krebs cycle, which is the oxidative phosphorylation. So it's not going to the mitochondria. It's not being turned into multiple ATPs. It's instead staying in the cytosol and being quickly turned into lactate and other byproducts of glycolysis. And in parallel, they saw an increased rate of lactate, saying that indeed there is more glycolysis going on. And then they also looked at this pentose shunt, which is a side biochemical pathway coming off the main one, and they looked at multiple genes in that and found that those products were also being upregulated, indicating that indeed sugar was being chewed up through this alternative pathway.
I thought that was particularly elegant because they actually queried the genome, and they said, are the genes responsible for this actually working harder?
Right.
And they were able to isolate all of the individual genes that would have been correlated with that switch and said, yes, they are working harder. And therefore, this is a consistent biological phenomenon.
And they did that in 2 ways, which I thought were really sort of rigorous. One was to look at these the genes on DNA are often tagged by methyl molecules, which are basically like little on-off switches that say, stop making this gene, whatever it is, this protein, whatever it is. And they found that the methylation was reduced, indicating that the genes were upregulating. Okay,
so cool.
It was so cool. And then they looked to see,
I have never seen the two of you so excited.
so the methylation, so the off switches have been removed to the on function now, and so we're making more of these genes. But can we prove that these genes are actually being expressed? And so they looked at the mRNA, the ribonucleic acids that's being messenger RNAs that are being created by this.
Boom!
And those went up too. So it's like,
Same
absolutely,
thing.
they are doing it. They are creating more of these enzymes that are involved in the pentose shunt and glycolysis. It was very, very elegantly done in terms of the basic science.
Right.
I thought I was really taken by this. I thought it was fascinating. I thought it was hard to read, and it made me sort of gulp and bite my tongue and go, "Wow, I forgot a lot of basic biochemistry," but it was still all there. Yeah, eventually.
And it was really interesting because the effect didn't seem to occur until 3 or 5 years later.
Right. Which is weird. But
Which is really
it makes
weird.
sense because BCG is such a weird vaccine, and it does not work the way most vaccines
Now, frankly,
do.
we don't understand how it works. We really don't. We really don't.
I feel like it's important for the listener to know that the way we're set up in the studio is Don and I are sitting across from each other with Chris in the middle. And the reason why you normally hear Chris's voice disappear is because he keeps turning his head away from the mic. And you will notice on that last segment, his voice didn't change at all. He was just talking directly to Don because I didn't get half of it.
Fair enough, man.
Let me also
Can we get back to the phosphoribose, please?
No.
I thought that was so cool.
Can
Enough with the pentose shut.
The pyrimidines.
Oh,
Can
come on,
I
stop.
give my my 2 cents here, which is not much because
You understood the first 3 paragraphs?
I understood the first 3 paragraphs. First of all, this is another one of those nature journals where the methods are at the end, which I
Haven't you learned by now? You have to go to the end.
don't get why you would do that.
It's like a mystery novel.
But the methods themselves are they're hard to follow.
Not well explained.
And I don't just mean that they are lab-based and therefore they're hard to follow. It's hard to follow the numbers. Where do these people come from?
It's the clinical
How many
part
of
that was
them-
hard to follow, right?
Yeah. And it strikes me that when I was reading this, that I was thinking to myself, this is really a lab study in a lot of ways. This was done in humans. There was a human component to it, but a lot of this was done in the lab, and it wasn't done, I always get confused, in vivo, in vitro. Which one was?
Vivo.
In vivo. And because of that, I feel like I'm supposed to view this with a different lens, that I'm not supposed to interpret it the way that I would interpret an epi study. And yet it strikes me as I still have to interpret this the way you interpret an epi study, largely because they make statements in here that I think are incompatible with this being a lab-only study. Things like, this should be something that we should be trying out in type 2 diabetes. Don't we need evidence that it really truly works in type 1 diabetes first? It's interesting, it's fascinating, but we're talking about a study in 8 people. In 8 people that I don't really understand where they came from.
Might have been 14, but
16.
right, whatever.
8
It's
and
a
8.
very small number.
It's a very small number. They were randomized to an intervention and a placebo, but with that many people, you really don't get much of a benefit to the randomization. And the small number followed up for 8 years is really, the 8 years is impressive. The small number makes it very hard for me to say that these benefits, particularly these benefits that didn't appear for 3 years until after the BCG vaccination, hard for me to know exactly what to make of this.
I totally agree with you, Matt. And actually, I have a very close friend whose name is Chris. Hello, Chris, if you're listening, who's a type 1 diabetic. And we were talking about this paper over the weekend. And he said, well, what I would really like to know as a type 1 diabetic is, do they provide any data about the decreased use of insulin? Because if their sugars are normalizing, you would expect the insulin demand to go down. And actually, they don't present those data that I could find anywhere in here, which struck me as sort of like, another example of what you're saying, that if this was a traditional epi study where we're really looking at clinical outcomes, that it would be absolutely one of the things that would be front and center as a surrogate marker of diabetes, control.
Yep.
Are you reducing your need for exogenous insulin?
Yeah.
And they don't present that, which seemed to me like, what a pity, because that seems so obvious.
Absolutely. I mean, this strikes me as a proof of concept study, not a conclusive study. And there is a statement in here that says the health benefits of the BCG vaccine reported here. So not the general benefits that we might postulate are out there. The health benefits of the BCG vaccine reported here suggest reconsideration of vaccine policy in areas of the world without endemic TB.
That's a bit bold.
Yeah,
Really?
That's a bit much.
That's a bit bold.
Let's I'm
reproduce the study.
uncomfortable with one large observational or intervention study determining policy. We have a study here of 16, let's say, people.
12 to 14. 12 to 16?
Okay, somewhere a very small study that I don't really truly understand the selection forces that were going on and how everybody got into this study at the end. I'm just not there. As fascinated as I was, I also part of it seemed too good to be true. So there is a lot of language in here that I just am so surprised that I don't normally read. So when they were talking about BCG vaccine in the introduction, so they weren't specifically talking about type 1 diabetes necessarily, although they were a little bit, they say that BCG vaccine halts MS, multiple sclerosis, halts. Is that,
I haven't looked at that literature, but they cited several articles, and it does seem to have an effect.
An effect would suggest that it reduces or probability, but this says
But if it halts it,
the
we would be doing
progression
it everywhere.
Halts the
It
progression.
implies that it's universal. They also say there are benefits to low birth weight infants independent of TB. And in mice, it permanently cures type 1 diabetes.
In mice.
Permanently cures.
Yeah.
That sounds like it
Yeah,
works
no, that's the
every single time.
Well, I don't know about every single time, but that's the regeneration of the pancreas in the mice.
No, it just strikes me as too early to be true, and then too good to be true. And then in this study, they found the same thing in that they said, well, I don't know, but they say that the lowering of blood sugars to a normal range near normal was maintained for the next 5 years. So it drops, goes back to normal after roughly 3 years and stays there for the rest of the time. It achieved these lowered blood sugars uniformly in all treated subjects.
All 6 of them.
Well, or 8 or whatever it is. But I'm just saying, like, even then, I'm just surprised it's every single one of them. That just, what treatments do we have that work universally?
Yeah.
Nothing.
Yeah. Cyanide.
Yeah, and vaccines work most of the time in most people, but I'm just
I think you're right, Matt, and I think it's an interesting sort of offshoot of an immunology lab, a very, very well-respected immunology lab, and bench scientists who are trying to anneal themselves with an ongoing clinical trial with limited numbers, admittedly. And maybe next time when they do it, they'll do it in a more comprehensive way, and they will then be able to at least provide the information to the reader from the clinical standpoint in terms of what's going on. I also do have to agree that this could potentially be really important in type 2 diabetes as opposed to type 1 diabetes, because it could, in fact, mitigate the need for any kind of a treatment whatsoever, because that's not a state of lack of insulin. This intervention does not address lack of insulin.
Yep.
It addresses how the cells utilize glucose. And that's essentially what happens in type 2 diabetes, because the cells become resistant to insulin, and they're much less efficient at being able to use glucose in the cells.
So can I end with, because we'll move on, with just one question, which is this was a mechanistic study at heart. They were really trying to figure out not just does this work, but how does it work? And the question that I have for you guys is, as fascinating that is, and I can understand absolutely why we'd want to know that, because that could lead us down all kinds of different pathways. Do we need to know that? Is it necessary? In other words, could we go out and just BCG is a vaccine that is approved for use in much of the world. Is there any reason why you couldn't just go out there and do a trial of, say, type 2 diabetes and see if it works? And maybe we never actually understand exactly why it works, but it works. That we would be interested in this.
You certainly could do it, but I think it's kind of the renaissance of BCG in the last 10 years, looking at all of these different groups, whether it's overall mortality or low birth weight mortality or multiple sclerosis, kind of implies that there is some overarching mechanism. And I think it's really, really important to get a handle on what that overarching mechanism might be. And this, I think, adds a lot to the argument that it is a profound immunologic regulation.
To my mind, this is where it was in some ways the weakest part of the paper, because the assumption had been, as you're saying, that the BCG is going to work in some way by modulating the immune system, because that seems to have been how it worked in mice, and that's why if this
And humans.
and that's why if this observation about multiple sclerosis, maybe not being cured but being remitting to some degree because of BCG, that has obviously got nothing to do with the Krebs cycle, right?
Yep.
We're talking about a totally different.
Yes, I have no idea.
Right, that's not about sugar metabolism. Multiple sclerosis does not, as far as we know, have anything to do with
Could be as far as I know.
aerobic glycolysis.
Yep, okay.
I think that would be a stretch. And so the problem is that they went into this study assuming that there would be an immunologic tweak that would mediate this restoration of pancreatic islet function, but they did not find that.
Well, they didn't make that final connection.
They couldn't make that connection, right? That's where it broke down, is that they actually, they showed that the T regulatory cells, which are involved in autoimmunity, are altered in a way that might lead to, create an environment that would allow the pancreatic islet cells to come back, but the pancreatic islet cells did not actually come back. And so when we're sort of thinking about what about type 2 diabetics? What about other conditions? This is where it gets a little bit dodgy, because we've walked down a pathway that actually doesn't necessarily lead to these other conditions.
It could be that it's true, possibly related.
Yeah. So further research.
More research is needed. And I think a lot of research, further research is needed. All right. I want to move on because in our second segment, we want to talk about something totally different. So have either of you done any research in your career that you now look back on and you are no longer convinced is correct?
Well, almost all of it.
Uh-huh. Yep. Chris?
I'm just trying to figure out
Maybe
whether I was
to make this
convinced to
simpler,
begin with.
do you have any that you're convinced is right?
I have can't even think about that hard. Convinced is right? Like beyond no.
No. Convinced is wrong.
Convinced is wrong. No, but I'm also open to the idea that there could be alternative explanations for things.
So what I want to talk about now is something that I came across on Twitter following folks that I follow in the psych research world, which is something called the what?
You follow people in the psych research world?
I follow a lot of people because there's some really fascinating things going on. In fact, a lot of the things we've talked about on this podcast have come out of things that have been going on in the world of psychology research, because they are going through this really interesting state of methods renaissance.
Interesting.
And so this is where this came from, which is something called the loss of confidence project. And so I think we've talked a bit on this podcast before about a lot of the big changes that have been going on in the psych world around questionable research practices, something we talked about, QRPs we talked about before. And more recently, there's been some very high-profile cases of research studies that were long taught as seen as the dogma that have now come up to be either problematic or falsified. So there's the marshmallow study. Do you all know about the marshmallow study?
No. What is that?
You don't know about the marshmallow study?
Tell us
This is
some
sort of a
more.
very famous study about that. I can't remember when it was done, the 70s or so in Stanford. It was a study that was done where you put kids in a room and you give them a marshmallow and you say, you can have the marshmallow.
Oh, yeah.
But if you wait, I can't remember how long they tell them to wait, like 10 minutes. If you wait and don't have the marshmallow and you sit there with the marshmallow in front of you, I will give you 2 marshmallows. And they found that the kids who could wait dramatically predicted the rest of their lives, their success in the rest of their lives. Only this was seen as dogma for a long time. And recently there was a replication study done, or I don't know if you call it a replication, but anyway, that it seems to be almost entirely explained by socioeconomic status of the mother that was not controlled for in the analysis. There was also the Stanford prison experiment, which is another one I don't know as much detail about. Anyway, there's all these things going on. And then, of course, there was the power posing that we talked about. And it gets to this idea of what do we do if we have done research that we look back on, and now that we've developed more sophisticated methods, we come to believe may not actually hold up. And that is the idea of the loss of confidence project. So it's been put together by Tal Yarkoni, Christopher Chabris, and later joined Julia Rohrer. And the basic idea is to destigmatize the idea of declaring loss of confidence, I'm reading from their website, in one's own research finding, in this case within the field of psychology. They're collecting statements of loss of confidence based on theoretical or methodological problems in one's research. Authors can submit a statement via their loss of confidence form. You can provide information about your study and why you think that it was no longer you have confidence in the findings. And then I think the idea is then they're going to put this all together and write a paper, including all the authors, to truly talk about this idea of how we destigmatize losing confidence in one's own research as one understands the methodologies better and comes to understand it. I thought it was a pretty fascinating idea. It's something that is not going on in our field. Mm-hmm. The question is, should it? Is this a good idea? And if so, do you think this is going to work?
I think it's a great idea. I don't know how well it's going to work, but
why?
I think it is. It takes a lot of guts to acknowledge that you believe something you published is wrong. I think that's hard to do. I think there's a lot of ego sort of wrapped around protecting or arguing for, advocating for your scientific findings. So I think that's going to be a huge barrier.
But I think I agree. I think it's a good idea, and if we can accomplish it, I think it'll be a real boon for the profession, because I think that when we are recognized as being as objective as possible, that'll enhance the confidence in the work that we do. Because if if new evidence comes up that your theory is no longer relevant, then I think it's really part of the basic progress of science to reevaluate what was proven before and change your mind about it if there's new evidence. And I think that's what they're getting at. I agree. They're really talking about those instances where your study could not be reproduced, even though you did it with the highest of integrity and the highest application of quality control. Sometimes it just doesn't happen. And rather than coming forward and saying there was something duplicitous that I did or something, somehow I cooked that's a different scenario. This is really as the context and circumstances evolve, the thing that I was bound to and adhered to previously is no longer correct.
Mm-hmm. So why not just retract the study? If that's the case, I mean, if we really have lost confidence in the research, why go to this step of having a place where people can put their hand up and say, I no longer have confidence in my research? Why wouldn't you just go the extra step of retracting the study?
Well, I think you could. I mean, I think that's not necessarily. I'm not sure whether they're proposing this.
They're actually not. I mean, they specifically sort of don't tackle that question in their FAQs. They say, one of the FAQs is, if I put something into the Loss of Confidence Project, will journals retract it? And they specifically say they don't know. We can't say.
But it's almost as if it remains in this database, it's really in a backwater, and it's not going to help our generalized knowledge of that particular issue. It really to become effectively diffused throughout the scientific literature, it should be retracted. If it's in fact found to be incorrect now and looked correct 20 years ago, 15 years ago, 10 years ago.
Maybe we should bring in the Cuddy-Carney.
Go for it.
So this is sort of the poster child in a way for this issue. This was a paper written by Carney, Cuddy, and Yap, I think.
That sounds right.
The third.
Yap.
And they were researchers in the psychology department at the University of Columbia, and they had investigated this theory.
Were they all at Columbia?
That I think at the time they were at Columbia, but they've all sort of since moved on to different institutions. So there was this theory that by adopting powerful stances, hands on your hips, sort of looking, jaw squared, looking forward, that this would not only make you feel sort of more powerful, but would actually lead to physiological changes manifested by increased testosterone levels and decreased levels of cortisol. And they measured these physiological changes before and after asking individuals to adopt power poses.
Chris is in a power pose now.
I'm not in a power pose. I'm jaw forward and looking steely like Bob Mueller. So
right,
we're not retracting nothing. So anyway, so this was.
Should
this
we have
was published.
A loss of confidence in this podcast?
And this
submission form,
this power pose paper, which had a sample size of 47, so it was a very small study, became a very big deal and led to basically launched the career of one of the three authors, Amy Cuddy, who went on to do a TED Talk, which has been downloaded 43 million times.
And wrote a book.
And is now a professor at Harvard and is like, this was this huge big deal.
She may be on leave from Harvard.
But the problem was that subsequently there was a much larger study by Rosenstein, I think, and they couldn't replicate it. It was the basic problem is they couldn't replicate it. And then a whole series of other follow-on PowerPose studies came out, and they could also not replicate it. And so Carney responded to this by basically saying, in light of what we have now learned, I think that we were probably wrong with the original study, and here are the reasons why I think we were wrong. And these included, among other things, p-hacking, which she acknowledges straight up that they started with a sample size of X, like 20 or 30 people, and then they didn't find anything, so they added on another 10, and they didn't find anything, so they added on another 5, and voila, they got a point of 5, and they published it. And so that would fall into our recurring bundle basket of questionable research practices, the QURPs, right? And so she talks about this, and she said, back in the day, everyone was p-hacking. We thought we were just being efficient, but now we realize that this is like doing multiple interim analyses and waiting until you get a hit before you publish in the New England Journal of Medicine, which we see as being kind of, Dodgy, right. And so she went through this process, and I thought that the mea culpa that she offered was really quite frank and forthright.
I thought it was excellent.
And it was excellent, yeah. And so in that I was like, wow, this seems a very legitimate way of sort of saying, this is what I thought, but now we see new data, and now I think something else.
So I agree with you that I think what she did was excellent. I think it reads, it's a very clear explanation of what happened and why she no longer agrees with it. The question is, was she in some way forced into this? And I'm not really so much interested in whether or not she was forced into it so much as it seems to me there was a big public controversy. She had to make a decision one way or the other. Was she going to support it or go against it? Whereas, it seems to me the loss of confidence project is not about the situation where one is forced to make a decision. It's about the situation where one comes to their own conclusion that I no longer have faith in my previous work, and I want to raise my hand even though nobody is asking me to. And I just wonder whether that's going to work. I mean, the example that we have in front of us is an example where you had to do something one way or the other. You had to make a statement of, I support my research, I totally disavow it, or I'm unsure. But most research isn't put into this light, and therefore isn't going to be forced to make a stand. And I wonder whether efforts like this are going to fall short because it's only going to be in the cases where people have a really bright light shown upon them that people are going to make these decisions to say, yes, I no longer support what I did previously. Right. And I just wonder if it's going to work.
Right.
Yeah, and if it's going to, if it has any hope of having the intended effect, as I said, there's got to be a link from the information presented in the lack of confidence website to to PubMed or something, so that that reference comes up.
Yeah, so I think we all agree, good idea in theory. Whether or not it's going to work in practice and have the intended, I shouldn't say I know exactly what their intentions are, but whether or not it's going to move the needle remains to be seen. We agree? All right. So then with that, let's move on to our third segment, our Amazing and Amusing. So this is where we want to highlight some of the things that make us love our jobs even more than we already do. I love my job, but this helps out a lot. Look at the weird and wacky things that happen in our field, according to Don, or the awe-inspiring things that happen to Chris. Who wants to go first? Chris, you ready?
Sure.
You look like you're warming up.
Sure. I'm just doing my stretches. I'm doing my power poses, in fact.
Yep.
So this is a little bit of an older study. It was published in 2007, so this is now 11 years old, but seemed really apropos given the theme today.
Okay.
Which is, the title is called Persistence of Contradicted Claims in the Literature by Tatsioni Benitsis and our man, John P.A. Ioannidis. And the genesis of this is to ask the question, how does the scientific community respond when a popular meme, and then in this case, these are memes that were generated on the basis of observational data, is subsequently refuted by a very large randomized controlled trial? And so the three examples that they talk about are, the belief that vitamin E supplementation is protective against cardiovascular disease. And there had been many I guess, decades, in fact, of observational studies that led to an overall estimation of a 50% reduction in cardiovascular mortality due to vitamin E. And then there were several very large randomized control trials that blew this out of the water and, in fact, showed that there's about a 20% increase in cardiovascular disease due to vitamin E. So the question was, how quickly do people stop citing those original data, including the meta-analyses of the observational study, and change their tune to say, well, in fact, it appears, based on this randomized control trial data, that the opposite is true. And so they looked at what's called content citation, meaning that they found the papers that had cited these original papers, and then went and found those actual citations to determine whether they were saying this is evidence in support of vitamin E preventing heart disease, or this is evidence against vitamin E. So, I mean, you can continue to cite the vitamin E meta-analysis from the observational data, but the question is, how do you represent it? Do you say this has now been discredited, or are you continuing to say it is true that vitamin E protects against heart disease?
Maybe we need a loss of confidence project.
It was very similar to that, in fact. And so what they found, and they didn't just look at vitamin E, but I'll focus mainly on vitamin E. They also looked at beta-carotene and the use of hormone replacement therapy for prevention of Alzheimer's disease. And both of those followed the same arc in that they had initially been assumed to be true based on observational data, followed by a large randomized control trial that showed exactly the opposite, and then how quickly did these sort of extinguish from being cited in support of that original theory. And so what they found in all three cases is that the scientific community does respond, but that the response is sluggish, that there, even in the years following, there's a long time before the alternative, the previous explanation, or the previous hypothesis is being rejected routinely in the scientific literature, and sometimes over a decade. And then there's a split in the kinds of journals that where the flip to the more recent interpretation occurs. And it's interesting because I thought this was very counterintuitive, that it is the general medical journals that seem to be more flexible and change their opinions quickly, like the JAMAs and the New England Journals and the BMJs of the world. They flip very fast in response to the new data, whereas it's the medical specialty journals that seem to be particularly stubborn in this regard, who hold on to the old beliefs. And they don't explain why that is, but one wonders if it's because these are where many of these epidemiologic studies originated, in the first place. And so now we're looking at a loss of confidence thing, or are people stubbornly holding on to their previous beliefs? And part of the strength of their argument that that's probably true is that following the vitamin E study and following the hormone replacement therapy for prevention of dementia study, there was a torrent of sort of counter arguments that were thrown up against the RCT to try to bolster the original observational data, which seems completely backwards, if we assume that there's a relative hierarchy in terms of things likely to be true in clinical medicine. Sort of like expressing that stubbornness, and nearly all of that backlash was coming out of the specialty journal editorials. It was really an interesting paper.
But I think that also there needs to be a time for absorption, discussion, and conclusion. And you say 10 years is a long time, but unless that the conclusive study is a complete slam dunk and just kicks out of the water all the other studies, there's going to be some debate and refinement. So that doesn't surprise me so much.
No, you're absolutely right. I mean, whenever I write a paper, I don't necessarily know all the things that have been published recently. So you're always a little bit vulnerable to missing something that just happened that would change your opinion. But it is striking how slowly this happened. So,
seems pretty slow.
Looking at the vitamin E, the pivotal study was published the RCT was published in the year 2000. So in 1997, looking at the citations and the favorable versus unfavorable, in 97, 77% were saying that vitamin E prevents heart disease, and only like 2% were saying it doesn't. In 2001, the year after the big RCT came out, those proportions changed to 57% favorable and 14% saying, no, it doesn't work. And it was only by 2005 that you approached a 50-50 mark in terms of the two sides roughly saying, yes, it does, no, it doesn't. So almost a decade.
To get to 50-50?
To get to 50-50. Oh, well, actually, it's 5 years. Sorry, it's because it came out in but still 5 years to get to almost 50-50, which is surprising.
That's pretty surprising.
Yep. So,
very cool.
Fascinating insight into our egos here.
All right. So I'm gonna go next, and I'll let Don Bat clean up, even though there are only three of us. So mine is intended to follow on to our first discussion of the study of BCG for type 1 diabetes, although this relates more to type 2 diabetes. And this one was actually sent in by a listener, Louise Tangerman. I apologize if I mispronounced your name, but this was something that she shared with me. And it came from Significance, which is the official magazine / website of the Royal Statistical Society. And this was an article titled Diabetes, Blood Sugar, and Red Wine, a Personal Study. And this was done by Martin Bland. Do you know Martin Bland?
I know the name.
He's a famous statistician, has done a lot of work with Doug Altman. He retired as I'm just going to read this part, professor of health statistics at the University of York following academic posts at St. George and St. Thomas Hospital Medical Schools. He was in industry, and he's co-authored almost 300 papers, many with Doug Altman. So he's a well-known, famous statistician.
He's a heavy hitter.
Very heavy hitter. And he developed type 2 diabetes and was managing his type 2 diabetes, and as a statistician was also then therefore collecting all this data on himself and starts to then track the information and starts to do some data analysis on himself. But then at one point, his wife, who he likes to keep informed of his sugar level, remarked one day that she thought that his blood glucose seemed to be a bit lower on mornings after he had had a drink. And therefore, he decides to do an analysis to figure out whether drinking in fact help him. So he does this analysis. He finds that there was a difference in mean glucose comparing red wine minus no red wine days equal to a small amount. I'm not going to read the amount. Therefore, and he's got a pretty good confidence interval around it, significant p-value if you care about that sort of thing. That therefore includes, thus we have good evidence for a relationship between red wine and glucose, which we estimate to be lower on average by between 0.06 and 0.51, millimole per liter on days after drinking. So a small effect, but the effect does appear to be real. He then sort of, writes this up, and the significance, he says, I wonder whether it would be feasible to do a trial to establish whether the short-term red wine effect was in fact causal, because of course there are all kinds of ways in this study you could have confounding. He said, in principle, I could randomize myself to drink on some days, but not on others. However, I think that this would be unlikely to work. I have a social life, and I am honestly not that dedicated to this particular search for knowledge. I suspect that I would be even less likely to be able to randomize others to drink or not to drink on given days. Perhaps there is an animal model. Like most nutritional risk factors, red wine and glucose is hard to study, as we well know. And then I will just end with his final thought, which is, for myself, I continue to record my blood glucose. I have added other data, the type of other alcohol, the number of drinks, and some information about food consumption and exercise. And of course, I shall drink red wine as usual. After all, it appears to be rather good for me.
That's awesome.
Which I thought was great.
It's really good.
Call that the Bland effect.
We'll call it exactly that.
The perfect N of 1 experiment. I love it.
Great N of 1 study.
All right. I mean,
it's great.
After two articles of such erudition, I feel like I need to bring the bar
Which bodily
down
function are we going to talk about today, Don?
a little lower. No, this is not a bodily function. So what I'm going to do is I'm going to...
You have a phone out. Are you going to play something for us?
No, I just forgot the reference,
good.
so I just need to read the reference off my phone. But
Happy
no,
emoji.
this is an article that I found from the psychology literature.
Oh, well done.
Your domain, Matt. Francis T. McAndrew and Sarah Kenke in New Ideas in Psychology published a paper on the nature of creepiness.
Oh, yeah. On the nature of creepiness.
Right. So they're saying that, the perception of creepiness is pervasive in a lot of different cultures.
Yeah.
But there's no
I'm sensing
creepiness
some of it right in this room.
There's no creepiness construct. We don't really know how to define creepiness.
We don't?
And what goes into the definition of creepiness. Why would one person perceive something as creepy and the other person wouldn't.
know.
They said there's no prior research, therefore this is a study exploratory in nature. They did snowball sampling through invitations to Facebook events created by researchers aimed at student, faculty, and staff at liberal arts colleges in the Midwest. And they posed a question to the subjects, which was, imagine a close friend of yours whose judgment you trust. Now imagine that this friend tells you that she or he just met someone for the first time and tells you that the person was creepy. And they asked a series of questions with respect to different they asked 44 different patterns of behavior, 21 different occupations, and a list of hobbies that people thought might be creepy.
Associated with this person?
That this person would admit to that they thought was creepy. And so 95% thought that creepiness was more likely to be associated with male gender than female gender, and that's both the men and the women.
That sounds right.
And probably the one that is at the top of the list is steering the conversation towards sex was considered to be creepy on first meeting this person.
that is creepy.
But the other characteristics
On first
were
meeting them,
that the
I would
person
agree.
stood too close.
Yeah, that's creepy.
The person had greasy hair,
Oh, yeah.
had a peculiar smile, had bulging eyes and long fingers.
Kind of like Nosferatu.
Yeah, kind of. Unkempt hair, very pale skin, dressed oddly, and licked his or her lips frequently.
Yeah, that always weirds me out when people do that.
And then
I feel like I'm 50-50 on this list.
yeah, it could be. The person would laugh at unpredictable times. And then as far as hobbies were concerned, it was that does seem to be a theme through all these podcasts.
I don't know what you mean, Don.
Yeah. The hobbies that were considered to be creepy were collecting hobbies. In particular, if the person admitted to collecting dolls, insects, reptiles, or body parts such as teeth, bones, or fingernails were considered to be
is creepy.
especially creepy. And the second most frequently mentioned creepy hobby involves some variation of watching, following, or taking pictures of people, especially children.
Yeah, that's
Was thought to be creepy.
That is
And
creepy.
bird watchers, of all things, were considered creepy by many as well.
bird watchers.
Bird watchers. Now, weird. Like, where would people be that they would see bird watchers and be creeped out by that? Or is it that there are people with
I don't
They
big
know.
just think
cameras
it's
hiding in the
voyeuristic,
woods?
I
What
guess.
are they doing?
You know, they're just like looking at birds in the trees during mating season. I don't know.
A lot of that rings true, and a lot of that makes me think I should go and reevaluate my own behavior. There's a few things on there that I
Maybe you should shorten your fingers.
I will just tell you the story. When I
Do you
was
collect pigeons?
no, when I was when I had my wisdom teeth taken out, they gave me sodium pentothal and Valium, and
Before
apparently
or afterwards?
I want to go to your dentist.
before. And apparently, and I don't remember this, I got up after the surgery and I was wandering around the doctor's office asking them for my teeth. I needed my teeth because I wanted to make a necklace.
I'd worry if I were you, Matt.
Wow.
So I don't know. Do I collect body parts? I
That's
don't know.
weird.
Wow. Okay. Well, that
So now we have a definition for creepiness.
is
That is actually a really cool study.
Take
Pretty
that
good.
to the subway.
that's pretty impressive, actually.
Because
Well done.
it's basically saying there is no definition, but when you see it,
you know
it.
when you see it.
Right. Well, now we have a framework,
I
a
like
creepiness
that.
framework.
That takes us advanced.
Yeah.
All right. Well, that is the ending of our program. So if you've got any feedback on this or any other episodes, or you want to suggest a study or a topic for us to take on, you can tweet us at @PopHealthyX, or you can tweet me at @ProfMattFox. Stop doing that. Or Chris at ID.Gill, or Don at See, what I'm most worried about is the listeners can't necessarily tell us apart by voice, and no one knows. I suppose they probably assume it's not me, but that's Chris, just so everybody knows. Where was I? We want to thank Leslie Talalian, Director of Lifelong Learning at BU School of Public Health, for supporting the podcast, and Nick Cooler for sound and editing. Thanks for joining us. We hope you enjoyed it, and we hope you download the next episode.